Good day to all of you. This video is about the Western blotting, and in previous video we have already discussed about the Northern blotting, which is used for the RNA specification. So in this it is used for the specific protein to detect the protein. So it is also called as the protein immunoblotting because an antibody is used to specifically detect its antigen. So principle of the Western blotting. So this is a technique consists of three major processes, that is separation of the protein by size, that is electrophoresis, transfer to a solid support, that is blotting, and the marking target protein using a proper primary and secondary antibody to visualize, that is detection. So here are the steps of the Western blotting. First, the extraction of the protein, then electrophoresis in the SDS page, blotting, blocking, treating with the primary antibody, treating with the secondary antibody, and treating with the specific substrate. And then detection is carried out by the help of the autoradiography or the fluorescence method. So in electrophoresis process, the SDS phase sodium dodecyl sulfate polyacrylamide gel electrophoresis is used for the electrophoresis process, and this SDS page maintains the protein in denatured form and reduces disulfide bonds, and the medium is negatively charged. So all the process is carried out by the linear structure of the protein. So for this, the disulfide bond has to be break. So then separation of the protein may be the isoelectric point, that is pi point, first dimensional gel, molecular weight, and the electric charge, or combination of these factors. Second step is the blotting. So electrophoretic transfer or the electroblotting is used for the transferring the protein from gel to onto a membrane and made up of nitrocellulose or the polyvinylidene difluoride. Then the gel is placed in a direct contact with the membrane. The different methods such as diffusion transfer, capillary transfer, vacuum transfer, electroelution are also available. So proteins are stained by the convention blue or the Pontian green, which is green in color, and then blocking process. The untreated site of the membrane is blocked by stopping the non-specific binding of protein probes to the membrane, and it is immersed in the BSA solution, that is bovine serum albumin, which is a 1 to 2% or the casein 1% or non-fat milk with some detergent. After that, the membrane is washed thoroughly and to remove any superfluous detection. Then the detection is held with the antigen antibody probe, and the enzyme, when exposed to its substrate, produces some detectable color. So the membrane is incubated with the primary antibody, which is specific to the target protein. And then the secondary antibody is also used, which is specific for a portion of the primary antibody is linked to a fluorescent probe, that is horseradish or peroxide, fluorescence, rhodamine, or the alkaline phosphate. The unbound probe is washed away and the signals are detected by the colorimetric method, the chemiluminescence, or by the fluorescence method. This pictures describe the Western blotting method. So firstly, the cells are centrifugated and the lysing the release of the cell protein. Then it is transferred into a tube, and the precipitate reaction goes on. Then the cell is fragmented and the collect supernatant, and then it is transferred into a SDS page where protein samples and markers loaded in vertical SDH page system. Again it is transferred into this membrane and then transferred into the BSA solution, the process known as the blocking process, which includes the two types of incubation. First, the primary antibody incubation, then the secondary antibody incubation. In primary antibody incubation, the primary antibody is attached with the probe antigen, that is again attached with the membrane. Then this in secondary antibody incubation, the secondary antibody is attached with the some specific part of the primary antibody. So and then when it is attached with this, the emission of the light, and this light is detected into the Western blot detection. So this is a figure of the Western blotting process. Applications of the Western blotting is used to determine the size and the amount of the protein in a given sample. It is used in the disease diagnosis and detect the antibody against virus or the bacteria in serum. It is confirmatory test for the HIV disease, HBV, and the HSV test. So identification of the specific protein in a complex mixture of the protein, and identification of a new discovered protein, isolation of desired protein, and is used to mark the gene expression in the cells after cloning and the recombinant cells. The demonstration of the specific antibodies in the serum for diagnosis of the meningitis and the neurocysticercosis, etc. So this is a whole about the Western blotting, their types, their methods and steps and principles and processes. Thank you.